Hello, my name is GabriÃ and I'm the first author of the paper entitled Fluorescently labeled vedolizumab to visualize drug distribution and mucosal target cells in IBD. In this video, I will briefly present the findings of our study published in Gut. Vedolizumab is a medication approved for treating inflammatory bowel disease, including both Crohn's disease and ulcerative colitis. It targets the gut and reduces inflammation. However, despite its approval, there are still significant gaps in our understanding. We don't yet fully know how vedolizumab is distributed within the body and which cells it targets. We also face challenges in determining the optimal dosage for individual patients, which is essential to maximize effects and minimize potential side effects. We investigated the use of fluorescently labeled vedolizumab to gain insight how vedolizumab is distributed in the inflamed gut, to identify vedolizumab potential target cells, and to assess vedolizumab target saturation. By determining how well the drug binds to its target, we can evaluate whether current dosing is sufficient. In this overview, I will describe our methods step by step. Firstly, we developed a fluorescently labeled version of vedolizumab according to GMP standards, known as vedolizumab 800CW. Next, we administered vedolizumab 800CW to IBD patients 2 to 4 days prior to their colonoscopy. In the third image, you see the in vivo procedure. During this procedure, we were able to simultaneously visualize high-definition white light endoscopy and the fluorescent signals in real time. Moving on to the ex vivo procedures, we first examined all biopsies for fluorescent signals to differentiate signal intensities between inflamed and non-inflamed tissue. Then we used the fluorescence microscope to perform a detailed microscopic examination of this signal. Additionally, we conducted 2 immune panels to identify which specific cells were bound by vedolizumab. Here I show a brief overview of our results. At the top you see the high-definition white light images from 3 different study groups: control patients, patients receiving 50 mg of vedolizumab 800CW, and those given a 300 mg therapeutic dose of vedolizumab, followed by 50 mg of vedolizumab 800CW. Below are macroscopic in vivo images from our fluorescence camera and biopsy images. We observe a clear fluorescence signal in both in vivo and ex vivo, with a notable decrease in signal after the therapeutic dose, as shown in the graphs next to the images. These findings indicate that our fluorescent labeling method tracks vedolizumab distribution and highlights the impact of prior therapeutic dosing. Next, we investigated the biopsies by using our fluorescence microscope to visualize the fluorescent signal of vedolizumab 800CW. In the control group, we detected no fluorescent signal. In the treated group, we observed a clear signal, but there was a significant reduction of the fluorescent signal in the group that had received pre-dosing with 300 mg vedolizumab. In this image you can see the overlap of the cell surface specific vedolizumab signal with plasma cell staining, suggesting that plasma cells may be a newly discovered target for vedolizumab. To a lesser extent, we also observed binding to macrophages and eosinophils. In conclusion, our use of fluorescently labeled vedolizumab has provided valuable insights into several areas. Firstly, we visualized the distribution of vedolizumab within the inflamed gut at both macroscopic and microscopic levels. Secondly, we identified potential and novel target cells for vedolizumab, finding that it mainly binds to plasma cells. Lastly, we assessed target saturation by analyzing signal reductions after treatment. Overall, these findings enhance our understanding of vedolizumab distribution and interactions, paving the way for more effective and personalized medicine in IBD.